So as biodiversity continues to decline, genomic tools are increasingly being used to improve conservation guidelines by safeguarding the adaptive potential of threatened species, with an emerging field being conservation epigenomics, specifically focusing on DNA methylation here, as it can complement traditional genomic insights by offering a layer of information that's tightly linked to the environment, with promising applications such as biomarkers of recent environmental exposures to guide a precision conservation approach. And sea turtles are a set of 7 endangered species where both genetic and epigenetic insights are really valuable for informing management under climate change, as they have a strong plastic link to their thermal environment as ectotherms with temperature-dependent sex determination, with near-complete female bias of sex ratios projected by the end of the century. So our paper showcases how Oxford nanopore technologies can be leveraged to simultaneously generate genomic and epigenomic resources, which is particularly cost efficient for species like sea turtles, where both types of data are useful for answering conservation questions. And to do this, we focused on the Northeast Atlantic loggerhead turtle population in Cabo Verde, which is just off the coast of West Africa and is now the largest loggerhead nesting aggregation in the world. And here we collected a blood sample from a wild nesting female on Sal Island in collaboration with the amazing local NGO Project Biodiversity, which we then sequenced via nanopore to achieve long reads for reference genome assembly. And after polishing and scaffolding, this achieved a high-quality chromosome-level reference genome assembly and annotation for this globally important population. And importantly, at the same time, we also profiled the blood methylome at over 26 million CpG sites directly from our nanopore reads, which we confirmed to be representative of the population via whole genome bisulfite data of an additional 10 loggerheads, with blood methylation profiles being really useful in a conservation monitoring context, as it can be sampled non-lethally and minimally invasively, while still reporting on a wide range of conservation relevant traits such as thermal exposure, sex, as well as health and age, just to name a few more. And finally, we applied our novel reference genome and methylome resources to yield a variety of insights relevant to loggerhead conservation. For example, at the population level, we linked past climatic shifts to effective population size fluctuations. At the chromosome level, we identified microchromosomes as special regions of functional genetic diversity and epigenetic flexibility. And at the gene level, we characterized nearly 200 genes linked to temperature-dependent sex determination, highlighting 3 potential interspecies rearrangements and building a map of their functional associations and methylation status to guide future study design, which you can read more about in our recent paper in GigaScience.